Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with the 12th standard biology chapter, that is human reproduction. So in today's video, we are going to learn the gametogenesis. So gametogenesis is the process of formation of male and female gametes in the testes and ovaries of the humans. Now it is of two types, that is in males it is called as spermatogenesis because the male gamete is the sperm, and in females it is called as oogenesis and the female gamete is called as ovum. So let us first see the process of spermatogenesis. Now in testes, the immature male germ cells called the spermatogonia produce the sperms by spermatogenesis, and this process begins at puberty. Now the spermatogonia are present on the inner side of the seminiferous tubules and multiplied by mitotic division and increase in number. So this is a diagrammatic view of the seminiferous tubule, and here we can see these are the spermatogonium which will divide mitotically and form more cells, and they will increase in number. Now each of these spermatogonium has 46 chromosomes. The spermatogonia forms the spermatocytes that undergo meiotic division to produce secondary spermatocytes. So these spermatocytes, the primary spermatocytes which were produced, they will again undergo division to form the secondary spermatocytes which will be haploid in nature, that is they will have 23 chromosomes. So these will have 23 chromosome number. After these, the spermatids are transformed into spermatozoa by the process called spermiation. Now after the secondary spermatocytes, the spermatids which are produced, they will be transformed into the spermatozoa or the sperm by the process of spermiogenesis, and these sperm heads will remain embedded in the Sertoli cells, as we can see over here. These sperm heads will remain embedded in these Sertoli cells, and they will be released from the seminiferous tubule by the process called as spermiation. Now let us see how the hormones play a role in the spermatogenesis. Now spermatogenesis is initiated due to the increase in secretion of a gonadotrophin releasing hormone by the hypothalamus, and the increase in this hormone, it acts on the anterior pituitary and it will stimulate the secretion of two gonadotrophins, that is the LH and FSH, that is luteinizing hormone and the follicle stimulating hormone. Now the LH will act on the Leydig cells which are present along with the Sertoli cells in the seminiferous tubules, and it will stimulate the secretion of androgen, and the FSH, they will act on the Sertoli cells and stimulate the secretion of some factors which will help in the spermiogenesis. Then talking about the structure of sperm. So as we can see here in this diagram, the structure of sperm is divided into a head, neck, a middle piece and a tail. So the sperm head contains elongated haploid nucleus. So here the nucleus is present inside the head, and the anterior part of the head is covered by this cap-like structure called as the acrosome. So this structure, cap-like structure, is called the acrosome. Then this acrosome, it contains some enzymes which help in the fertilization of ovum. So when the sperm comes in contact with the ovum, the enzymes which are present in the acrosome will help with the fertilization. Then the middle piece of the sperm possesses numerous mitochondria which produce energy for the movement of tail and facilitate the sperm motility. So this is all about the spermatogenesis and structure of sperm. Now let us see the oogenesis, that is the how female gamete is produced. Now the process of formation of mature female gametes is called as oogenesis. Now it started Now this oogenesis process is started during the embryonic development stage, when millions of oogonia or the gamete mother cells are formed in each of the fetal ovary. So this process basically starts when the baby is in the embryonic development stage. Now the gamete mother cells start division and enter into prophase 1 of meiotic division, and they will get arrested there. Okay, they will get temporarily arrested there. And at this stage, they are called as primary oocytes. Now each primary oocyte gets surrounded by a layer of granulosa cells, and it is called as a primary follicle. So from the embryonic development stage till reaching to the puberty, from these millions of oogonia, a lot of oogonia get disintegrated or destroyed, and at puberty only about 60,000 to 80,000 primary follicles are left in each of the ovary. Now the primary follicle gets surrounded by more layer of granulosa cells, and it is called as secondary follicle. So after puberty, the primary follicle which is there, it will again get covered by more granulosa cell layers, and it will be called as secondary follicle, and then it will again transform into tertiary follicle, which is characterized by a fluid filled cavity called the antrum. Now the primary oocyte which is inside or within the tertiary follicle, it grows in size and it completes its first meiotic division to form a large haploid secondary oocyte and a first polar body. Then the tertiary follicle further changes into Graafian follicle, and after that it ruptures to release the secondary oocyte or the ovum, that is the egg from the ovary, and this process is called as ovulation. So hope you have understood this. Thank you for watching my video. Please like, share and subscribe to the Bodhi Academy.